I'm Dr. David Wikenheiser. I'm a naturopathic physician, and I work with people to solve their health concerns using regenerative medicine. This video is the fourth in my series on Low Dose Immunotherapy, LDI. This is an important therapy for balancing your immune system. In this video, I will discuss how low dose immunotherapy works to reprogram your immune system and give you back tolerance and balance in your immune reactivity. So low dose immunotherapy works by stimulating the production of what are known as T-reg cells or thymus regulatory cells. So if we need a boost in our immunity, we've just got an infection, we need help, your thymus T helper cells will jump into action. Now, your thymus gland also produces T regulator cells. These are very important in our hyperreactive modern world, where many people are overreacting to immune stimulation or even reacting cross-reactivity, molecular mimicry against wrong tissue. In other words, they're attacking themselves and vigorously. This is not a good health circumstance. So let's give you an analogy. If you drive a car, you know that when you push down on the gas pedal, you'll go forward. Your motor will rev up and you'll have more energy and you'll go forward. Why, when you take your foot off the gas pedal, do you slow down? Now, in the most simple terms, why that happens is because there's a spring. And when you take your foot off the gas pedal, that spring pulls, if you've got a gas-powered car, it pulls the carburetor closed and you don't get as much gas and air into the engine. So there's a spring that down-regulates the operation of your motor. So in our bodies, that spring that closes the carburetor in our car, in our immune system, that spring are T regulator cells, thymus regulator cells. Molecular mimicry is the overreaction of a body against parts of our body that the amino acids, the proteins, look like a bug that they were attacking. In the case of rheumatic fever, heart disease, that would be strep bacterium in the throat infection that your immune system reacted against, and then part of the protein structure of that bacterium looks like it's molecularly mimicking the structure of the heart valve. So your heart valves look like the strep bacterium, and then you get cross-reactivity, you get your immune system attacking the wrong thing. So we've got misdirected immune system and a lack of ability to calm it down. So when we produce the T regulator cells, we regain our ability to calm down our immune system. So how does this actually happen? We have dendritic cells in our immune system. Now, if you may have heard of dendrites on nerve cells. These are the multi-branched structures at both ends of a nerve. They're at the beginning to pick up a nerve impulse from other nerves, goes down the body of the nerve, down to the end. So there's dendrites that put out containers, little boutons, buttons for neurotransmitters to be emitted to go to the next nerve. Those types of receptor dendrite cells, they're similar structures in our immune system, and they pick up and sense what's in our environment. So if we are able to take a trigger, an immune antigen, something that's going to trigger our immune system to react, if we can represent that to our immune system, to the dendritic cells of our immune system, then we can reset our immune system. Now, you may have heard of dendrite and dendritic cells as part of cancer therapy to get the immune system to retune and go after cancer cells. So similar to that, we use with low-dose immunotherapy a reprogramming of the dendritic cells with low dilutions, so low concentrations, high dilutions of the triggering antigen. So for example, if someone is highly allergic to wheat, we would dilute, so the dilutions are 1 part per 100, and that's known as a 1C. You shake that up, stir it up vigorously, and then you take a portion of that and you do 1 part of that into 99 parts of sterile water, and you successively do that out. Now for foods, that might be anywhere from a 6 to a 12 C. For problems such as Lyme infections, Lyme disease infections, Borrelia burgdorferi, that can be as much as 32 serial dilutions of 1 in 100. So we take a trigger, dilute it out to the point where you're not going to react to it other than to make T regulator cells, present it to the immune system through dendrite cells at a low enough dilution to get the immune system to react, and then it'll downregulate. This is really an interesting thing, and it works. It's quite amazing. So we represent an immune trigger to the body reprograms tolerance. You avoid the trigger for a week after low dose immunotherapy, so it's really drops, and we figure out which dilution is best for the individual under their tongue. And this is drops under the tongue, it's not injections or other immune type therapies. So drops under the tongue, your mouth is full of dendritic cells, the immune system is sensing what's there. You reprogram the immune system with a treatment, you wait a week, and then challenge. So if you had a food treatment with hopefully treating wheat, you can challenge and see if you're forming T regulator cells after a week. Now if you're highly reactive and really possibly anaphylactic with a food, then you might just put the food on your skin, or don't put it in your mouth, put it on your lip. Just do a minor exposure and see if in fact you're making enough T regulator cells to calm down your immune system and not have it overreacting. So that's how low dose immunotherapy works. It reprograms your immune system to calm down the immune system by making T regulator cells, by stimulating their production. They'll last for many months. And over time, as you build up T regulator cells, in 1 to 2 years, you may not even need treatment after that. It will take some time, but not forever. So we're going to go into video number 5, the history of low dose immunotherapy. I'm Dr. David Wikenheiser, and here we go with my 5th video discussing low dose immunotherapy.